All right, welcome to the Explainer. Today, we're diving into a classic topic that you are guaranteed to see on your board exams, thalassemia. This is a core red blood cell disorder, and really knowing the key differences between the types is absolutely crucial. So let's jump right in. Okay, so picture this. It's a classic clinical question. You've got a patient, they have microcytic anemia, small red blood cells. You do the logical first step, you check their iron, and it's totally normal. So what's jumping to the top of your differential diagnosis list now? Yep, this is the exact setup that should make you think thalassemia.
So what exactly is thalassemia? Well, at its heart, it's a genetic disorder where the body just can't make enough of the globin chains. Those are the proteins that build hemoglobin. And here's the absolute key, the thing you have to remember. This is a quantity problem, a reduction in synthesis. It's not a quality problem where the chains are built wrong. You're just not making enough of the building blocks.
To really wrap our heads around this, let's just quickly review what normal adult hemoglobin, we call it hemoglobin A, is actually made up. It's what we call a tetramer, which just means it has 4 parts, 2 alpha chains and 2 beta chains, all fitting together in a perfect balance. The entire problem of thalassemia kicks off when the production of one of these chains gets all out of whack.
And that imbalance leads us down 2 main paths. You've got alpha thalassemia, where you're not making enough alpha chains, and the genetic defect for that is on chromosome 16. And then you have beta thalassemia, where, you guessed it, you're not making enough beta chains, and that defect is over on chromosome 11. Let's break down each one of these.
All right, first up is alpha thalassemia. And if there's one word you need to burn into your brain for this one, it's deletion. See, the issue here isn't a faulty gene, it's a missing gene. Every person has 4 alpha genes in total. We get 2 from each parent on chromosome 16. The severity of the disease, from being completely asymptomatic to being fatal, depends entirely on how many of those 4 genes are just gone. And this table here, it lays out the whole spectrum perfectly. If you're missing just 1 gene, you're a silent carrier. Your blood work looks totally normal. If 2 genes are deleted, you get what we call thalassemia minor or trait, just a mild microcytic anemia. Now, things get serious when 3 genes are gone. That's hemoglobin H disease, a much more severe chronic anemia. And if all 4 are deleted, the fetus just can't make functional hemoglobin. This leads to a condition called hydrops fetalis, which is sadly incompatible with life. Okay, exam time, let's hit the buzzwords. For populations, think Southeast Asian and Chinese. When you're missing alpha chains, the other chains that are still being made have to team up, and they form these weird abnormal groups. So excess beta chains form hemoglobin H, and fetuses, excess gamma chains form hemoglobin Barts. And here it is, the absolute key for diagnosis that trips people up. In the common alpha thalassemia trait, the hemoglobin electrophoresis is normal. Okay, so let's switch gears and talk about beta thalassemia. Remember how alpha was the deletion story? Well, this is the mutation story. Right, so instead of whole genes just being gone, the problem here is usually a point mutation within the gene itself. These mutations can either lead to a reduced amount of beta chains being made, we call that beta plus, or they can cause a complete absence of beta chains, and we call that beta 0.
And this, again, creates a whole spectrum of disease. If you're heterozygous, meaning you have 1 bad gene, you've got thalassemia minor, which is pretty mild. If you're homozygous for one of those milder beta plus mutations, you get thalassemia intermedia, and you might need occasional transfusions. But if you're homozygous for the severe beta 0 mutations, you have thalassemia major, and this is a very serious transfusion-dependent condition.
And the buzzwords for beta, you gotta know these. Think Mediterranean populations, so Italian, Greek. The most severe form, thalassemia major, also has an eponym, Cooley anemia. Now, because the bone marrow is just working on overdrive, trying to pump out red cells, it expands like crazy, leading to classic bony deformities. And the huge complication from all the transfusions needed to treat it? Iron overload. All right, so we've got the theory down. But how do you actually figure this out in the clinic? How do we nail the diagnosis and tell it apart from its biggest lookalike, iron deficiency? First up, the big showdown, thalassemia trait versus iron deficiency anemia. They're the top 2 causes of microcytic anemia. Now, both have a low MCV, but in thalassemia, it's often super low, like disproportionately low. But this is the money right here, the RBC count. In iron deficiency, you can't make the cells, so the count is low. But in thalassemia, your body is churning out tons of tiny little cells, so the RBC count is paradoxically normal or even high. And, of course, the iron studies are normal. Okay, so you're pretty sure it's thalassemia. Now what? How do you tell alpha from beta? The key test, the one that tells you everything, is hemoglobin electrophoresis. Like we said, in alpha thalassemia trait, it's normal. But in beta thalassemia, the body's trying to compensate for those missing beta chains. How? By ramping up production of its other hemoglobins, like hemoglobin A2 and hemoglobin F. Seeing those elevated levels is the smoking gun for beta thal.
So when you actually peek at the blood smear under a microscope, what are you going to see? Well, you'll definitely see microcytes, those small cells, and hypochromia, which means they look pale because they're lacking hemoglobin. You'll also see a really classic finding called target cells and something called poikilocytosis, which is just a fancy way of saying the red cells have all sorts of weird shapes.
But wait, there's one specific finding, a high-yield buzzword that should make you scream beta thalassemia, and that's basophilic stippling. When you see these little blue dots sprinkled inside the red blood cells, that is a massive clue. Now, when the disease gets really, really bad, like in thalassemia major, the bone marrow is under such incredible stress that it just starts spitting out immature red blood cells way too early. So when you start seeing nucleated red blood cells on a peripheral smear, you know the process is severe and that bone marrow is just in overdrive. Okay, last big piece of the puzzle, management. What do we actually do for these patients? How do we treat them, from the really mild forms to the most severe? Well, it's really all tiered by severity. For the minor or trait forms, you do nothing. Seriously. The main thing is genetic counseling so they understand the risk of passing it on. For the intermediate forms, we do supportive care, give them folic acid, and make sure they avoid things like iron supplements. For thalassemia major, the absolute cornerstone of treatment is lifelong, regular blood transfusions. And because of that, you have to manage the complications, which means using iron chelation therapy to pull all that excess iron out of the body. And now we're even seeing curative options like stem cell transplants or brand new gene therapies.
So after all that detail, if you walk away from this with only one single concept, please let it be this one. Thalassemia is a problem of quantity, not quality. The body just isn't making enough of the necessary parts to build healthy hemoglobin. That one idea explains everything else we've talked about.
And that really brings us to the future, which is kind of mind-blowing when you think about it. For literally centuries, having thalassemia major meant your life was tied to blood transfusions. But now, with these revolutionary gene therapies becoming a reality, you have to ask, are we on the edge of a future where we can actually fix the fundamental genetic problem and free these patients from transfusions for good? It's a really exciting time to be in medicine.